Okay, so this video is going to be looking at glycolysis and the important regulatory steps, the important regulatory enzymes, and also the obviously the byproducts of glycolysis. Glycolysis starts. Glycolysis starts at the top here with glucose. Now glucose is a hexose sugar. It is polar, but it has no net charge. However, it can't diffuse through the phospholipid bilayer. However, it is able to pass into the cell from the blood by some transporters that are known as the GLUT transporters, and these are very important because depending on whereabouts in the body the cell is, there will be a different expression of the GLUT transporters. So GLUT4 is insulin sensitive, and also have some of them like GLUT1 and 3, which will be present more in the brain, etc., that sort of thing. But anyway, so the glucose is uptaken into the cell. Now, to prevent the glucose from then diffusing back down out of the cell, down the gradient, if there such a gradient were to exist, like negatively outside from inside the cell flowing outside, the glucose is phosphorylated by hexokinase, and that allows the addition of glucose, a phosphate group, to the glucose on the 6 carbon, forming glucose 6-phosphate. Now this is an important step because it involves the investment of a ATP molecule, and ATP then can go on to form ADP and PI, and the ADP will be free in the cytoplasm, and the PI will bind onto the glucose. So why would this be done? Why would this be done? Let's just mark that up as a question mark. What is the significance of phosphorylation of glucose? Phosphorylation of glucose decreases the concentration of glucose within the cell. This will mean that more glucose can then diffuse. The net diffusion of glucose into the cell will increase. This is also an energy investiture. As everyone knows, ATP is the universal energy currency of the cell. So if you are putting ATP, if you're using ATP to phosphorylate, then you can get this energy back at a later point during glycolysis. Also, the putting on of a negative phosphate group onto the glucose stops the diffusion out of the cell. So this enables you to keep the glucose 6-phosphate within the cell. The next enzyme to act on the glucose 6-phosphate in this step is a isomerase, and the isomerase allows the production of the fructose 6-phosphate. And all this does is change the conformation. This is not a highly regulated step, so it's not important to really know about that. All you need to know is that there is a change in the conformational structure of the glucose 6-phosphate to form fructose 6-phosphate. However, the next step, which is carried out by phosphofructokinase, which is another kinase, so we're adding another ATP. Just arrow that in here. So we also have the addition of ATP here at the top, and also here at fructose 1,6-bisphosphate. So phosphofructokinase phosphorylates fructose 6-phosphate and leads to the formation of fructose 1,6-bisphosphate. This is a highly regulated step. So phosphofructokinase is regulated by a number of cellular components. So ATP will cause a downregulation. So lots of ATP means that you don't need any more ATP. So that will downregulate allosterically the phosphofructokinase. ADP or AMP will do the opposite. So high energy signals downregulate, low energy signals upregulate. Now, one of the upregulators for phosphofructokinase is fructose 2,6 bisphosphate, and this is formed from the fructose 1,6 bisphosphate. This is like an isomer of, so that will downregulate that, because if it's formed from the fructose 1,6 bisphosphate, then that will downregulate it, because that will indicate there is a high level of fructose 1,6 bisphosphate. Now, the next step in the glycolysis pathway, I'll just underline the fact that we've just changed colour and underline the fact that we've just jumped through the first major part. There is a division of fructose 1,6 bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And these two components act in dynamic equilibrium between each other. So dihydroxyacetone phosphate can go on to form glycerol, which is an important part of cellular metabolism in the formation of triglycerides, but it will form glycerol phosphate, and then it will lose the phosphate and be able to form glycerol, where it can travel freely around the blood. Glyceraldehyde 3-phosphate, on the other hand, will undergo a series of reactions, where it will then go on to finally form pyruvate at the bottom of the glycolysis. There are a number of other steps as well which are quite significant. So the most significant one to remember is that there is a liberation of ATP and the production of NAD (2H). And NAD2H is a hydrogen carrier which enables the energy to be utilized in the body. Pyruvate can then go off into the link reaction, where it will become acetyl-CoA. It can be turned into lactate. So there you go, acetyl-CoA. There you go, lactate. And also it can go on to form various other metabolites within the body. So you can use it, it can form dynamic equilibriums between some of the components of the TCA cycle, so like oxaloacetate and malate, and also amino acids. It's also important to note that there is a key regulatory step, which is pyruvate kinase, which is situated right there. And pyruvate kinase will remove a phosphate group in the from the pyruvate from the phosphorylated pyruvate. So this kinase actually works in the opposite way around to your normal, and that will then produce pyruvate, and that's a high energy step, so that also has to be regulated as well.